Hi, class, and welcome to Chapter 12, which is on DNA technology and genomics. This will be a brief lecture presentation. It's an extremely long PowerPoint slide, but I'm also going to put a YouTube video in the modules for you to take a look at as well. So a little bit about an introduction and why this is important. Duchenne muscular dystrophy is a fatal genetic disease caused by a mutation in the gene that encodes dystrophin, which is a protein vital to muscle development. Researchers have used a harmless virus to deliver gene editing components to the muscles of dogs, and within weeks, normal dystrophin levels were restored, providing that Duchenne muscular dystrophy could be halted in a large mammal. In addition to medical applications, DNA technologies can affect our lives in many other ways. So we're going to take a look at your DNA and genetics, and we're going to ask the question, are genetically modified organs safe? We'll look a little bit about gene cloning and editing, genetically modified organisms, DNA profiling, which is what we'll go over that probably specifically because that's what your discussion post will be about, and then genomics and bioinformatics. So gene cloning and editing. Gene cloning is one application of biotechnology, which is the manipulation of organisms of their components to make useful products. And researchers can manipulate bacterial plasmids so that they contain genes from other organisms. These recombinant DNA plasmids can then be inserted into bacteria. And if the recombinant bacteria multiply into a clone, the foreign genes are also duplicated and copies of the gene or its protein product can be harvested. So this is a look at glowing aquarium fish produced by transferring a gene originally obtained from a jelly. So now you can buy fish that glow in the dark. So this is just a look of an overview of gene cloning. It takes you through the long description in the notes below, but basically we locate a plasmid, the plasma is cut out, and then the cell's DNA is cut with the same enzyme from the cell with the DNA containing the gene of interest. And then the targeted fragment and plasma DNA are combined. DNA ligase is added, which joins the two DNA molecules. And then the recombinant plasmid is taken up by that bacterium. The bacterium then reproduces, and then the gene has the then clone is in it. So this is kind of a long process in the description of how gene cloning is. The following slides take you through each step individually of how, for example, gene cloning exists. So I would encourage you guys to go through these slides one at a time to kind of go through that process on your own. All right. So here's a great animation as well that shows the cloning of a gene. And especially for this chapter, because it is so long, the animations will provide a really good kind of summary of the cloning of a gene, as well as other things that we'll go over. Restriction enzymes cut the DNA at specific sequences, forming restriction fragments, and then DNA ligase will paste the DNA fragments together. So here's a restriction enzyme, how it will be cut, and then DNA ligase will put it back together into the gene of interest to form what we call recombinant DNA. Nucleic acid probes can label specific DNA segments. So for example, a short single-stranded molecule of labeled DNA called a nucleic acid probe can tag a desired nucleotide segment. Complementary DNA can be used to identify the genes that are being transcribed by a particular cell at any given moment, and that is shown here: DNA in the nucleus, mRNA in the cytoplasm, adding reverse transcriptase, synthesis of the new DNA strand, breaking it down, and then synthesis of a second DNA strand. The CRISPR-Cas9 system allows researchers to target the specific gene in a living cell for removal or editing. And that is shown here, how the Cas9 guide can pick out a specific gene that they want to clone, find the target gene, break it apart, and then add it to the final nucleotide sequence to be cloned. Genetically modified organisms, or GMOs, we've heard a lot about. Bacteria, yeast, cell cultures, and whole animals can be genetically modified to make products for medicinal and other uses. So, for example, a goat carrying a gene for a human blood protein that is secreted into its milk is shown here. Researchers can use these DNA technologies to produce drugs, diagnose diseases, and also produce vaccines, which are harmless variants or mutants or derivatives of a pathogen. So here's a look at human insulin produced by a bacteria and equipment used in the production of a vaccine against hepatitis B. Scientists have produced many different varieties of GMOs, which are organisms that have acquired one or more genes by artificial means. If a gene is transplanted from one organism into another, typically of other organisms, the recombinant organism is called a transgenic organism. And a number of important crop plants are genetically modified, as shown here. This is show how the TI plasmid is genetically engineering plants. And genetically modified organisms in terms of plants are important for creating a crop yield that is resistant to insects or other bacteria. So this is golden rice, which gives it the yellow color. Scientists are also investigating the potential risks to human and environmental health posed by DNA technologies. Why is it often necessary to run both human and animal studies to learn about human health? The animal studies give us a good kind of comparison of what could happen to humans. For example, here's the percentage absorbed and converted to vitamin A in a capsule of pure beta carotene, golden rice, and spinach. So these are genetically modified organisms to help kind of carry more vitamins. Gene therapy is changing a defective gene to a normal one in a living human. This shows promise for curing defective genes, but the actual success rates are rare. Why does bone marrow make a good target for gene therapy? And that's because it can easily transmit its therapies into the cells that are formed. DNA profiling. So this is what your final discussion post talks about. DNA technology is the uses modern laboratory techniques for studying and manipulating genetic material. It has rapidly transformed the fields of forensics, which is the scientific analysis of evidence for crime scene investigations and other legal proceedings. DNA profiling can determine whether two samples of DNA came from the same individual. So what it does is it takes DNA from a crime scene from two different suspects, and it's selected, its markers are amplified, and then in order to see the DNA, we compare it with each other or the three different samples in a gel electrophoresis. So what happens here is we're able to compare DNA across suspects. So we take DNA from the crime scene and the lines will match up with the correct suspect. So according to the data presented, which suspect left DNA in the crime scene? So suspect 1 or 2, and we're able to tell which suspect matches the DNA at the crime scene by seeing which line markers are most evidently lined up horizontally. And you can see here that suspect 2 has more lines that match up with the DNA at the crime scene. So suspect 2 is the one who committed the crime. The PCR can be used to amplify a DNA sample. Specific primers will flank or follow the same horizontal arrangement as the desired sequence, ensuring that only a particular subset of the DNA sample can be copied. So starting with a minute sample, it only shows that a particular subset of DNA can be copied. So again, this shows how different cycles get to subsequent cycles of horizontal DNA segments. So eventually, again, it's just explaining how the lines that match up more closely together will be the suspect. Many DNA technology applications rely on gel electrophoresis, which is a method that separates macromolecules based on size or electrical charge. And this is where we get to the bands of gel electrophoresis of DNA, each consisting of thousands of DNA fragments of the same length. So we put a big mixture of DNA fragments of different sizes. We get different horizontal lines showing. So the shorter, faster molecules will travel all the way down. And this is what it looks like under a black light. So eventually we can compare crime scene DNA to suspect DNA. And the ones that match up the most will be our suspect. The applications of DNA profiling can be used to solve crimes, establish paternity, so who's the father, and also identify victims. In what ways is DNA profiling valuable for determining innocence as well as guilt? Well, it can also rule out people. So here's the source of the samples, semen, the markers, and which that exonerated Earl Washington. So Earl Washington obviously didn't match up as well as the other suspect, so he was exonerated, so it proved his innocence. Genomics and Bioinformatics, I'll let you guys read through this a little bit on your own. This is next and third generation sequencing machines that can quickly determine the sequence of relatively short sequences of DNA, as shown here. And these large machines, genomics researchers have sequenced many prokaryotic and eukaryotic genomes. Besides being of interest in their own right, non-human genomes can be compared with the human genome to show how genomes are similar through different species, as shown here. And I'll let you guys read through this a little bit on your own. The Human Genome Project was a massive publicly funded scientific endeavor to determine the nucleotide sequence of all the DNA in the human genome and identify the location and sequence of every gene. So this is really cool. That happened several years ago. Data from this project reveal that the human genome contains just under 21,000 genes and a huge amount of non-coding DNA, much of which consists of repetitive nucleotide sequences. So here's a look at the repetitive DNA, the introns, the non-coding DNA, and the repetitive DNA. Modern genomic analyses then can depend upon the whole genome shotgun method, which involves sequencing and arranging many small DNA fragments at the same time. So this is a look at a chromosome, the DNA fragments, how we can align the fragments and reassemble the whole sequence. Bioinformatics is the use of computational methods to analyze biological data. This can be used to analyze large sets of data about DNA sequences and proteins. Proteomics involves similar systematic studies of the full protein sets encoded by genomes. So here's a look at the alignment of nucleotide sequences of the Malabi leonina, which lacks the deletion found in the other 4 species. So through this, we're able to basically see the genetics of every species. Using databases like GenBank, we can compare genome sequences across many species to look at some of the relationships between these species being tested. So this is just really neat to kind of compare different species. Here is a long list of things you should be able to do from this chapter, but I would encourage you. This is the man who was exonerated by the DNA profiling. Reviewing concepts, DNA profiling. This is a great picture to study and what words you can kind of insert to review there. And that takes us to the end of chapter 12. Thanks for listening.